University of California. And with that, okay, Irene, you have the Zoom screen.
All right. So the first thing I want to do is thank everybody for their interest in coming to this, because I have to say that I've been doing quite a number of these talks, and I do them because I need a goal in order to do a lot of deep diving into research to do the presentations. So I thank you because it's fun for me. So thank you very much. So Joe will be sharing with you a YouTube that I made early this year after I'd done a few presentations and thought, you know, it really just needs to be recorded and set out. And it will give you, I hope, a very solid background about the mRNA vaccines, the nature of the virus, the nature of the cellular response to the vaccines that protects you in the future. And a very important point, which is the only way to stop variants is to get vaccinated. And we'll talk a little bit about that. So with that, I'm going to share with you a slide which for me ties together all the pieces of what I'd like to find time to present to you today. The YouTube will cover background and information about the 50 years of research that went into the vaccines. They didn't come out of nowhere. Where did they come from, and what's the nature of those vaccines? And at the end, if we have time and you have interest, I'd like to talk to you a little bit about a big detective game that affects all of us that's going on right now, which is, where did this virus come from? Was it a product of laboratory research, or was it something that came out of the woods through various intermediate hosts and happened to gain the ability to infect humans? So that is what we could get to. So what ties all of these parts together is what's called gain of function. So gain of function is a term that we use in genetics when an organism, you know that evolution is a completely random process, and therefore many of the changes that happen genetically in you, in me, in viruses, in bacteria, all of those changes are random. Most of them are garbage, and they just kind of drift out of existence, or they get carried silently in genomes. Gain of function genetic changes are different in that they acquire a new ability. It's like when a baby learns to walk, that's a gain of function. When a virus that lived in a bat gets the ability to live in a human, that is a gain of function change. So every variant that has been generated during this pandemic is a variant of concern only if it makes our disease more lethal or more morbid. That would be a gain of function change. Viruses go through all kinds of random changes that are invisible to most people, except those who are trying to sequence, to track where gain of function mutations are coming from. The current variant of concern is Delta, right? That is a gain of function variant. Billions of changes happened, and that one happened to be a gain of function that's causing us problems. So I will put up a slide for 1 second just to show you kind of that trajectory. So again, gain of function, sorry. So gain of function. So as I said, variants of concern, the things that we talk about in the news, those are gain of function. And I put in parentheses, loss of function variants. They're lost in the noise of sequencing. Vaccines are the only way to protect us against gain of function variants, those that make this disease or any disease worse, any virally caused disease worse. The research that has been in the news quite a bit, done in China, centered in the city of Wuhan at the Wuhan Institute of Virology, was done in good faith, funded by the United States, among other places. Gain of function research is the only way that the people in China could see where there was potential harm and be able to develop vaccines to head off a pandemic. Unfortunately, obviously, we have a pandemic and we did not have vaccines. It hadn't gotten far enough. We can go over that later. So that last bit that if we have time and you have interest, we can talk about is the question that I've put at the bottom of this slide. Was the gain of function of SARS-2, that's the shorthand for this virus, SARS-2, effected with or without lab effort? So that's the end of that. And we can go to either the share screen of the YouTube.
All righty, here we go. Click to share sound. And now I will bring up the video.
This video is for the general public. I'll explain how the current vaccines work and introduce you to the virus and the variants that are causing concern. I'm Irene Kuhn. I'm a cancer cell biologist. I earned my doctorate in Biochemistry and Molecular Biology at UC Davis. During my career as a research scientist, I've used many kinds of viruses to make vaccines and therapeutics. One of those viruses, an oncolytic adenovirus, is currently in clinical trials for the treatment of colon cancer. So viruses can be amazingly useful, but they can also cause havoc. Since the pandemic began, I've been reading scientific publications and listening to updates from virologists, immunologists, epidemiologists, and medical doctors. I've distilled what I learned into this presentation and invite you to share it with others. So let's get started with this talk about the COVID-19 vaccines, the virus, the variants, and immunity. The image is of the virus that is responsible for this pandemic, and you can see why it is called a coronavirus. The pandemic has been tough on all of us, very tough, especially on those who became ill or lost loved ones, and on the people who risked their lives to care for us. When businesses closed, many lost their livelihoods. When schools closed, family stress increased. And overall, our social inequities shamed us. Here's what I'll cover. I'll focus first on the mRNA vaccines and the 40 years of research behind them. I'll discuss the efforts to ensure racial and gender balance in the clinical trials. I'll explain how vaccines generally teach our immune systems to protect us, and then summarize the studies of how well the vaccines are actually doing. The next section is about the virus, what we've learned about how this respiratory virus spreads, how it infects and takes over our cells, how it makes new, potentially more dangerous variants. And I'll conclude with why reaching herd immunity through vaccination is so important. These are the kinds of questions I began with and that I'll answer here. I wondered why I should get vaccinated. Were the vaccines safe? What about how quickly these mRNA vaccines were made and approved? Could the vaccine make me sick? Could it change my DNA? Could it kill me? Let me get that one out of the way right away so you don't have to worry about it. No one has died from being vaccinated with these vaccines. No one. If I were vaccinated, what could I do safely? And why do I still need to wear a mask? So you can follow along. These are the basic terms of this pandemic. SARS-CoV-2 stands for Severe Acute Respiratory Syndrome-Related Virus. That's the SARS part. It's a coronavirus, CoV, and it's the second one of this type, number 2. COVID-19 stands for Coronavirus Infectious Disease 2019. mRNA is the acronym for messenger ribonucleic acid. Spike is the big protein on the outside of the virus that pulls the virus into cells. This protein is required for infection. Looking at the graph on the left, the red bars indicate the proportion of deaths due to COVID-19 in each age group, while the black lines indicate the proportion of the U.S. population in that same age group. Death from COVID-19 is more likely the older you are. In the graph on the right, the left column shows the proportion of deaths from COVID-19 for each racial group, and on the right, the proportion of the U.S. population for each racial group. Clearly, the non-white U.S. citizens are paying a heavier price in this pandemic than those of us who are white. I got vaccinated once I had become comfortable that the vaccines are safe, that they will protect me from getting seriously ill or even dying of COVID-19. I also got vaccinated to protect other people, because becoming immune by vaccination is the only way to stop the creation of new and potentially deadlier variants of this virus that would prolong this pandemic. So how do vaccines protect us? Vaccines teach our immune systems to recognize infectious agents, such as the SARS-CoV-2 virus, illustrated in the upper right-hand corner of this slide. This recognition by the immune system is very specific knowledge. It's like having a wanted poster of the culprit in fine detail. That specific knowledge is stored in your immune system's memory cells. Later, if you get infected, those specific memory cells very quickly reproduce and travel through your body to find and destroy the infectious agent, thereby reducing the possibility that you could pass the infection to others around you or get sick yourself. Memory cells are very long-lived, so they can protect you years later if this specific infectious agent appears again. We don't yet know how long the SARS-CoV-2 memory cells will be functional. We can only learn that as time passes. But we are encouraged as we now know that memory cells from the 1918 flu lasted at least 90 years. There are three parts or elements of good vaccines. The first is the message they send to your body. The second is the language in which that message is written. And the third is the delivery vehicle. It took over 40 years of research to figure out how to make excellent mRNA vaccines. I don't expect you to read all this, but I list it here to show that the research, the published research, on which the mRNA vaccines are based, goes back more than 40 years. The idea of these vaccines was carefully developed and tested and retested over many decades by many different, often competing groups of research scientists. Most of this research was funded by your tax dollars, supporting research groups at universities and national laboratories, such as the Infectious Diseases Division of the National Institutes of Health, and that includes the Vaccine Research Center. Also contributing to the great speed with which these vaccines became available to us is the unprecedented level of collaboration of scientists and research groups from around the world and global collaboration between governments and philanthropists that came together to fund companies so that they could produce millions of doses of vaccine candidates even before these vaccines had been proven effective. The damage of the pandemic engendered unheard of cooperation. Let's look at a few of the scientists whose decades of research resulted in the mRNA vaccines. We'll start with the vaccine now called the Moderna mRNA vaccine. As indicated by the photo on the left, the Moderna mRNA vaccine is actually a product of our National Institutes of Health's Vaccine Research Unit. You'll probably recognize Dr. Fauci and the former president, and to his right is Dr. Corbett. Dr. Corbett heads the mRNA COVID-19 vaccine team at the NIH. Dr. Kariko, pictured here, is one of the Hall of Fame scientists of this pandemic because of her 30 years of tireless work against great odds, trying and finally succeeding to make RNA vaccines work. Why was this work so hard? Because mRNA is very fragile. mRNAs are important molecules in every cell in your body. mRNAs encode the information that tell your cells to make the proteins required for life. And to regulate all that information, these mRNAs have to be destroyed as soon as they have delivered their instructions. So your cells rapidly eliminate mRNAs as soon as they are read. Because mRNA is so fragile, it took Dr. Kariko decades to figure out exactly what kind of mRNA bases could survive long enough to be useful, and how to purify that mRNA in the lab so that it would elicit the desired kind of immunity. And then how to encapsulate those mRNAs in microbubbles that could be effective vaccines. Here's Dr. Kizzmekia Corbett's team at the NIH Vaccine Research Center. Dr. Corbett is the lead scientist of the strategy she calls the, quote, prototype pathogen approach to pandemic preparedness, unquote. Using this strategy, she and her team have for many years been making a whole range of vaccines against those viruses that we know are most likely to cause the next pandemics, and then evaluating immune responses and protections against those same viruses in animal models. It's dangerous work handling all those viruses, and it's also complex and laborious process of test, adjust, repeat. Successful vaccines are then tested in humans once they are safe. Over the years, this group has developed mRNA vaccines that had entered early phase trials in humans. These include vaccines for rabies, Zika, flu, cytomegalovirus, and respiratory syncytial virus. I asterisk the RSV, or respiratory syncytial virus, because it was the intensive research by the next two scientists that was critical to the ultra-rapid design of the COVID-19 vaccines. The blue arrow on the left points to Dr. Barney Graham, chief of the Viral Pathogenesis Laboratory and co-director of the Vaccine Research Center at the National Institutes of Health. Dr. Graham is surrounded by his research team. Dr. Graham has focused on developing a vaccine for respiratory syncytial virus for over 30 years. Respiratory syncytial virus is a common infection, but can, unfortunately, cause severe disease and even death in young children. Dr. Jason McClellan, in the photo on the right, is particularly interested in the structure of proteins. He joined Dr. Graham's lab about 10 years ago to work on the structure of the RSV spike protein. Resolving the structure of a huge metastable or floppy glycoprotein like the RSV spike requires very high-powered cryo-electron microscopes and a lot of computer power. Together, these two scientists resolved and, in 2013, published the RSV spike protein structure, and also, importantly, figured out how to stabilize that huge protein in what is called the pre-fusion conformation. That is, the shape this protein is in on the surface of the RSV virus when it infects human cells. This version of the protein is called spike 2P. When Chinese scientists figured out and published the whole genome of the SARS-CoV-2 virus on January 10th, 2020, Dr. Graham saw immediately that the spike protein of this new virus, the SARS-CoV-2, was very similar to the spike protein of RSV. That was a huge breakthrough. Between them, when the pandemic struck, these scientists had all the experience needed to rapidly design an mRNA vaccine. Drs. Graham and McClellan knew exactly which version of the spike protein should be the message. Dr. Carrico had published the language and a variety of nanobubble delivery vehicles that could be used for mRNA vaccines. Dr. Corbett's years of testing showed how to put the SARS-CoV-2 vaccine together, message, language, and delivery vehicle, to make the most effective, immunogenic, highly protective vaccine. The clinical trials of the vaccine were designed to test the efficacy of this vaccine to protect against serious and lethal COVID-19. Over 30,000 people who were at risk, that is, likely to be exposed to the virus, volunteered for the trials. The trials had to be effective for all races and genders in our population. Equity and inclusion in the clinical trials is critical. Much work continues to be done to gain the trust of people of color, as these groups have been abused by the medical and scientific communities. Review and management teams for the clinical trials included BIPOC scientists and doctors, ensuring that racial and gender balance for vaccine clinical trials reflected the racial and gender balance of the United States. The Biden-Harris transition team, working in parallel, included experts from underrepresented groups. All of this is reflected in the next slide. This is a screenshot from an NAACP virtual town hall program available on the NAACP website. Note that Dr. Patrice Harris was president of the American Medical Association in the year before and during the pandemic. Dr. Nunez Smith, on the top far right, served as co-chair of the Biden Coronavirus Advisory Board. Dr. Corbett from the NIH was part of this panel of experts. She's shown on the bottom row, second from the left. So how well did the vaccine do? This is a page from the New England Journal of Medicine publication of clinical trial phase 3 outcomes. I've enlarged the graph on the right. The cases of serious COVID-19 illness in the placebo group kept rising over the 10-month trial period, while the yellow line shows very few cases in the vaccinated group. We have to say that adverse events are expected from any vaccination program, and we know that there have been a few. But we also know that COVID vaccines cause fewer adverse events than the flu vaccines that we get yearly. The next slides will look deeper into the Moderna vaccine, what it is and how it elicits a protective immune response. First, vaccine cannot make virus in you. The COVID vaccines can only make spike. Here's a cartoon of the SARS-CoV-2 virus. You'll notice those red things that look like golf tees sticking out of the virus surface. Those are the spike proteins we saw a few slides ago. The gene for spike is shown as a red coil, and it is the mRNA encoding spike that is in the mRNA vaccines. Notice that the gene coding for spike is only 4,000 letters or bases long. The whole genome of this virus is over 30,000 bases long, and all those 30,000 bases are required to make a virus. Since only 4,000 bases are in the vaccine, there is no way that the virus can be remade in your body. The message for spike cannot recreate the whole virus in you. The vaccine, encoding the mRNA for spike, teaches your cells to make copies of the spike protein that look just like the ones on the virus. That process is illustrated on the right. The vaccine is shown as the red coil of spike mRNA inside of the oily nanobubble that delivers the mRNA to your cells. These oily bubbles fuse with the fatty surface of your cells, and the mRNA enters the cell. The mRNA is taken up by ribosomes that read the mRNA instructions to make spike proteins. The spike proteins end up on the surface of your cells, ready to train your immune system. Once the vaccine is in you, when immune system cells, such as the B cells or T cells, bump into your cells that have newly made spike proteins on them, the B and T cells recognize the spike as foreign and they learn to defend you against the SARS-CoV-2 virus. The B cells learn to make thousands of different kinds of antibodies that can glom onto any COVID viruses that may get in you, and the T cells learn to destroy any infected cells. This process takes about 2 weeks, so please be sure to wait 2 weeks after your last vaccine shot before you consider yourself immune. This slide is a reminder that T cells are not only a very important part of our immune response, but they are also a very durable part. The new variant viruses may be able to evade some aspects of our B cell-made antibodies, but are very unlikely to escape the killer T cells. In summary, B cells neutralize virus particles while T cells kill infected cells. All the COVID-19 vaccines teach your immune system to make thousands of kinds of antibodies to attack the SARS-CoV-2 virus and create swarms of T cells that kill any cell that contains virus. Importantly, vaccines teach your body to create both B and T memory cells. The populations of these cells will quickly expand so they can attack any future SARS-CoV-2 viruses that infect you. Next, a look at how well these vaccines are working. Clinical trials measure vaccine efficacy, but the real test is how well do the vaccines work in a general population. This is effectiveness. Israel was the first country to have vaccinated a large percentage of its population. They recently reported results of their study in which they tracked the health of 1.2 million people, half of whom had been vaccinated and half of whom had not been vaccinated. The groups were balanced for age, gender, and pre-existing morbidities. Note that 30% of these groups were over 60 years old. The results show that the mRNA vaccine made by Pfizer was 94 to 96% effective at preventing serious disease across all age groups, even those over 70 years old. This shows the vaccine protection does not diminish with age, and it's such good news. It is particularly assuring that the vaccine performed so well in spite of the study being done during the largest outbreak of COVID-19, during which the more infectious United Kingdom variant had become the dominant strain in Israel. The next 2 studies of the possibility of reinfection after vaccination were done on staff at 2 medical centers. Both studies used the PCR test so that they could detect asymptomatic carriers as well as those with apparent illness. The LA group tested their vaccinated staff weekly. You can see the numbers of new cases dropping, see the second column from the left, as time post-vaccination increases. The last line is 15 days post-second dose, so full immunity would be expected. And you can see that 7 people out of 15,000 people, that's all, were either ill or found to be carrying the virus asymptomatically. The Dallas medical staff reported 4 people were either ill or shown to be carrying the virus out of over 8,000 staff tested. This percent of protection is in agreement with the LA results. Both studies show around 98% protection. These studies show a very high degree of protection from mRNA vaccines. The Pfizer-BioNTech mRNA vaccine encodes the same version of spike, the spike 2P, as the Moderna vaccine, but in a slightly different oily nanobubble. The Johnson & Johnson and AstraZeneca Oxford vaccines are both adenoviral vector DNA vaccines, and Sputnik V from Russia is very similar. So again, these vaccines encode the same message, the virus's spike protein, but using DNA as the language instead of mRNA. The DNA is delivered inside of attenuated adenoviral vectors that are taken up by your cells. The outer shell of the vaccine is stripped off inside the cell. The DNA enters the cell nucleus where it is transcribed into spike mRNA, which leaves the nucleus. The rest of the story is the same as for mRNA vaccines. The spike mRNA is translated into protein. The new spike proteins migrate to the surface of the vaccinated cell, where your T and B immune cells encounter the new spike protein and start learning to mount an immune response against this viral protein. This type of adenoviral-based vaccine has been used before to fight Ebola and Zika virus outbreaks. The safety and efficacy of these vaccines have been shown. These adeno-based vaccines have some advantages over mRNA vaccines. Only one shot is needed to become immune. This vaccine is stable in a refrigerator for 3 months. The one and done and refrigerator shelf life make these vaccines excellent for use in rural clinics and in emergency rooms where many people receive their health care. Note that these adenovirus cannot replicate and they cannot integrate into your DNA. To summarize the vaccines section of this video, all these vaccines are destroyed in your body very quickly. None of these vaccines can change your DNA. They all encode the same protein, spike 2P. They use either DNA or mRNA as their language, and they use different delivery vehicles. People have voiced concerns about side effects. We know that there will be side effects. That is expected for any vaccination program. But experience with previous vaccines shows that any side effects caused by a vaccination will have been seen within the first 2 months after completion of the clinical trials, so we can be prepared for them. It is this knowledge that supported the FDA's decision to release the COVID-19 vaccines 2 months after the completion of their clinical trials. The release of the vaccines was not rushed. That said, the CDC continues to collect data on everyone who is vaccinated. You can contribute to this effort by signing up on the CDC website for the vSafe program. The uplifting news is that the current vaccines protect you 2 weeks after completed vaccination very well against even mild COVID-19 and apparently completely protect you from dying of this disease. Next, let's look at the SARS-CoV-2 virus. Here's my favorite representation of the virus. The spike protein reminds me of fried chicken drumsticks. To start with, viruses are not alive. Viruses must infect us in order to make copies of themselves, in other words, to replicate. When the pandemic started, we were all very fearful because we did not know how this virus was spread. How could it infect us? Some viruses can remain infectious on surfaces, that is, other kinds of viruses. So we sanitized everything, doorknobs, packages, mail, food. We stopped shaking hands. Luckily, tests soon showed that this particular virus is one that does not survive very well on most surfaces. So we could relax a little bit about sterilizing surfaces. But please continue washing your hands, because if you touch your face, you may get something into your eye.
Irene, could we
One
take a
scientist
pause for a
isolated
moment
virus
to have a
respiratory
question?
virus.
Joe,
Common illness.
Can we have a pause? Thank you.
hello? Yeah?
Yes, I've paused it.
Right. Yeah, we have a question from Audrey.
Sure.
Why is the Delta variant more infectious than the first variant, and can it produce more severe effects in children?
I don't know about the difference between adults and children. I haven't looked into that, but I will cue you at the end, and Hillary will send out a link to a presentation tomorrow that focuses on the Delta variant given by UCSF Department of Medicine Grand Rounds, and that should be a very interesting session. So you'll get more information from them. But let me go back to the first part of your question, Audrey. Why is the Delta variant more deadly? And that goes back to the theme at the beginning that I talked about, gain of function. So when you have a viral outbreak, and viruses have different viruses have varying abilities to mutate and how quickly they mutate. We have to get vaccinations every year for flu because flu is one of the more mutation competent viruses. SARS is less so. That's a blessing. But when you think about gain of function, you can't just jump from step 1 to step 10. You need to go through all the intermediate steps. And so whatever Delta has acquired, it has acquired it on the shoulders of previous variants of concern. Does that kind of give you an idea, Audrey, about why is Delta more dangerous and why did it take so long to come out? I'm not hearing Audrey. Did that take care of your question?
Yes, it does. But I'm wondering if it was many mutation steps that had to occur.
Yes.
In order
Yes.
to appear.
Yes, very many, very many.
Wow.
And if we get to it, if you look at the phylogenetic trees, that is how each virus that has been fully sequenced is related to another one, it's incredibly complex. You would use up many computer screens looking at all of it. It's a whole study in itself. But yes, they have to kind of go in sequence, and that's been a concern. Yeah.
Wow. Well, thank you.
And Roger also had a question.
Roger?
Maybe a broader question. How does our experience with this pandemic help us prepare for the next one? And I might kind of tag onto that and say, why didn't the first COVID-1, COV-1 kill us all before the next one came along?
So there are, there's a wonderful article that came out in the New York Times, I think, right before I left to go into internet dark zone, which had to do with humility. Humility in the face of viruses. There is an enormous amount that we don't know about pandemics in general. And one of the things that epidemiologists and virologists are tracking right now is why were the outbreaks that we had so ferocious within certain populations and didn't seem to touch other populations? Case in point would be India. For a long time, people looked at India scratching their heads. India is very densely populated. Nobody's wearing a mask, and yet they did not have the kind of devastation that was occurring in the United States, especially in New York City, for instance. But then all of a sudden, bam, India got hit very, very hard. And we still don't really understand why India got better at the point that it did. It was not necessarily human intervention that stopped the virus. So we still don't know what's going on with this particular virus, and every country and every social system seems to be slightly different. So I don't know if that answers the question, but it's not something that's simple to answer. And but the self-attenuation of viruses is something that we have to be incredibly thankful for. There was the SARS outbreak in the early 2000s, followed not long by MERS. If you remember, that was a camel-derived virus that broke out in the Middle East. It was 30% lethality. That's huge. SARS is below 1%, it's 0.2 or something like that, percent lethality. MERS was 30% lethality. You really didn't want to catch it. And it attenuated very quickly. We still don't know exactly why. But thank goodness that it did. So as far as preparing for the next pandemic, that is a function, I feel, of humans. You know, we get through a tough spot and we don't want to spend the rest of our lives focusing on never having that tough spot again. It's fun to go off and do other things. And I think we forget and we don't pay enough attention to all the things we would have to do to avoid another pandemic. And it includes things like not expanding the human population, because it is the interface of humans and biodiversity that allows these viruses to come out of the animal reservoirs in which they are endemic and attack us. That's a very short answer to a complex question. Is that good? We're going on?
Yeah, would you like me to go on now?
I think so. Yeah.
All right, I'll do that. Just give me a minute here. It's the first time I have experience in pausing it here. It's going to take me a second to figure out how to do that, please. There we go.
Such as colds and the flu are caused by respiratory viruses, but this one is deadlier. Respiratory viruses are expelled from our noses and lungs in aerosol sprays or droplets when we talk, sneeze, or cough, or sing. We soon learned we can get infected by someone who is not sick. Because this virus takes a few days after we are infected before we feel sick, and during those few days, we are already contagious. This is a great photo showing the aerosols and droplets that spew out when we sneeze or cough. But note that his Kleenex or cloth stops most of the spray. That's why masks are so important. Wear them whenever you are out. You never know when you or someone around you might be contagious. This drawing shows two people standing close enough that aerosols and droplets emitted while speaking are landing on the other person. Social distancing keeps the spray zone away from you. Also note that your chance of getting infected while talking increases with the length of time you talk. Try to be brief. That's what it looks like by electron microscopy if your lungs become infected. The hundreds of virus look like spiky balls. It's not too pretty. A single virus infecting a cell can replicate 1,000-fold, releasing hundreds of infectious virus into your lungs. When you talk or cough, you can too easily infect the people around you. For a closer view, these images are from an animation showing how the SARS-CoV-2 virus infects cells. There's the virus in the upper left panel. This time the spike proteins are shown in green. The surface of the cell is below the viruses, looking like a gray rug. The ACE2 receptor, the one that the spike protein is looking to latch onto, is shown in purple, sticking out from the surface of the cell. Moving to the upper right panel, a closer view, here the spike proteins have found and latched onto their ACE2 cell receptor landing pads. What happens next is truly amazing transformation of the spike protein into a nanomachine that deploys tendrils down into the cell surface, pulls the virus surface smack on top of the cell surface, causing these two surfaces to fuse. Once fused, the virus shoots its genome into the cell. The bottom right panel shows the cell after the virus has entered it and moved toward the center of the cell. But note that the virus's spike proteins are left on the surface of the cell. Remember that vaccination teaches your immune system T cells to recognize infected cells and kill them. So the viral proteins left on the surface of this infected cell mark it for destruction. To sum up this brief section on the virus's spike proteins are the nanomachines that grab onto the cell surface and then pull the virus into the cell. The virus then takes over the cell, turning it into a virus factory. The virus replicates, producing thousands of virus. Viral surface proteins remain on the outside of the cell so that once you become immune, your T cells recognize those viral proteins and kill infected cells. But it is that massive replication of viruses that worries us, because that is when variants are created. Some variants have made the nightly news because they are more transmissible and/or they cause more serious illness. These variants are very rare, but every one of those variants was created when someone was infected, including possibly someone who is asymptomatically infected. This is true because viruses mutate, which is to say that they generate variants only when they replicate, and viruses replicate only when they infect someone. So as long as people continue to get infected, new variants will emerge. We worry that the vaccines we have may not protect us if the variants become so different from the original virus that our immune defenses cannot protect us from the new variants. Vaccines prevent infection. If a virus can infect, it can't generate variants. So that's how you can help stop the creation of new variants, by getting vaccinated. This point is so important that I will repeat it. Viruses are not alive. They cannot reproduce on their own. Viruses cannot produce variants on their own. Viruses are created only when viruses replicate inside our cells. That is, when we are infected. So when we get infected, the virus creates variants. Vaccinations reduce infections, reduce replications. When the virus cannot replicate, it cannot produce variants. Vaccines stop variants. Until you can get vaccinated, please stay safe and wear a mask so you don't get infected. As I am preparing this video, some people are vaccinated and immune and others are not. So here's how the CDC asks that we manage this. When fully vaccinated people are with vaccinated people, they can take off their masks and can have meals together, even indoors. If any unvaccinated people are in the group, masks need to be on to protect those who are not yet vaccinated. If no one is vaccinated in your group, or you don't know if there are unvaccinated people in the area, please keep all safe practices, masking, keeping distance, and frequent hand washing. These guidelines change as the pandemic changes, so check with your favorite news sources to keep up to date. New variants raise fears of increased transmissibility of the virus. Many people advise to wear 2 masks to double our protection against aerosols. So here's someone wearing 2 masks. Obviously, 2 masks completely overlapping. I won't read these off, but these are the resources I've used in preparing this video. They are public sites where you can get updated information. I particularly love the podcasts of the 5 or so scientists who broadcast This Week in Virology, affectionately known as TWIV, on Microbe TV website. Another wonderful web-available set of videos are those from Grapevine Health. These are the brainchild of Dr. Lisa Fitzpatrick. Dr. Lisa talks with people in the streets in her Ask the Doctor series. That's it. Long, but you've reached the end of this video. I hope you're more comfortable with the science behind the COVID-19 vaccinations. These vaccines are safe and effective, reducing severe disease to zip. It's amazing. Current data are looking very good, indicating that vaccination nearly wipes out transmission of the disease. Vaccination also nearly wipes out the chances that you can get reinfected by this virus if you are vaccinated. Obviously, we do not yet know how long this protection will last. So do not throw away your masks. Remember the shortages from last March? Masks are precious, especially after you've found one that fits your face. And on a final note, variants. Please remember that the only way to stop variants from cropping up and prolonging this pandemic is to get vaccinated.
That's it.
All right, I'll stop the share.
Thank you. All right. So do we have any lingering questions that we should deal with before we go on to a little bit more? And we're almost at 4 o'clock here, so.
Yeah, we do have a question from Elizabeth that refers to that study I mentioned to you this morning that was done by the Mayo Clinic. That the Pfizer vaccine has a coating of BioNTech, but the Moderna doesn't. And this more recent information, which is from Mayo, reported by Reuters, on how effective Moderna and Pfizer are against Delta after some weeks or months have passed. And Moderna scored higher than Pfizer. Moderna 70% and Pfizer somewhere around 45%. So could this be related to this coating that these two vaccines have?
You know, I am not deep enough into any of this to know how to answer that question very clearly. But I would say absolutely, that would be my first thought, is that they do have different nanobubble structures. They're both nanobubbles. They're both very well informed by all of the research that's gone into them. But it could be that that's what's going on. But it would take a vaccinologist, of which there are many, and I'm sure they'll start weighing in or have weighed in about why one of them would be 45% and another 70%. But I'd also encourage you to tune in tomorrow at noon to the UCSF program, because Sean Crotty is the featured speaker interviewee on that. And he has done an amazing job in working with all the different variants and immunity, the questions of how it's elicited, where it's elicited, who's responding, who's not responding. So for a deeper dive, join us at the link that I sent to Hillary, and Hillary will share with everybody at noon tomorrow. It's also available by 6 or 7 p.m. tomorrow night. You can access it any time. That link is available to you from Hillary.
Okay, that's in the chat now if people want to copy it.
Fantastic. All righty. So shall we go on to a little bit of detective work and see what about that? So let me carry you forward unless there's any urgent questions right now. Let's go on.
Oh, was a question from Celia.
Oops, let me do a stop share then. Celia, yes.
Yeah. From news reports, it seems as though breakthrough infections are becoming more common. What does this mean for herd immunity? If fully vaccinated persons can catch and pass on the virus, how does a high vaccination rate stop the spread?
That's a so the whole question of what will constitute herd immunity has been on the drawing board since the beginning of this pandemic. I'm sure you've all heard, even before the vaccines came out, people talking about what's sufficient vaccination to constitute herd immunity and how protective will it be. And I don't think that's a question that anybody that I've read has ever said, this is the answer, right? I think we are going through a common experience of this pandemic and our protection against it. And we're having to roll with a lot of different punches. and we have to be kind of flexible. There is no right answer to all of this. This is part of why I really liked that New York Times article a few weeks ago about the humility in face of the virus. We don't know perfection. The background that I gave you during the YouTube is, I was listening to it for the first time in a couple of months just now, and there are complexities behind each simplification that I put into that YouTube. It's a background YouTube. And I don't know that I would ever believe anybody who said, this is exactly how it is, and this is what's happening. I wouldn't believe it. So we have to get comfortable. This is part of the trauma of this pandemic, is we have to get used to the idea that there's something quite frightening around and we don't totally understand it. We understand a little bit. And part of why I made the YouTube was because I was so impressed with the rapidity with which a lot of research came together to rapidly get those mRNA vaccines out. I've worked a lot with adenovirus. I wasn't as impressed with that because it's more standard, but the mRNA has been phenomenal. And there's a whole world of, for instance, cancer vaccines that are right on the heels of those mRNA vaccines for COVID-19 that are very interesting. There's a lot of stuff going on. So let's just leave it at that, unless there's more questions. Okay, all right. So I'm going to do a share screen and just whip through a few slides. And what I want to say before I start is that I pulled together quite a lot of publications and research about the question of where did this virus come from? And the question is, as you know from the news and President Biden's decision to open a formal investigation, has a lot to do with our security, the question of whether or not some people believe, and for political reasons have pushed the idea that the SARS-CoV-2 virus, when it originally emerged, was either a lab accident or an escape from a laboratory experiment, SARS-CoV-2, and that it was being developed as biowarfare. That is one extreme view of where did SARS-CoV-2 come from. At the other end are a lot of people who have spent their careers looking for the next pandemic, and they look for it in the sense of analyzing thousands and thousands and thousands of viruses that are out there in the animal world and evaluating them for the potential to become human threats. So we have, on the one hand, this was intentional modification, and it had, at the worst, a malicious intent. And at the other end of the spectrum, we have, this is how nature works. There is a lot of crossover. And I have to say, as many of you know probably, our human genomes carry lots of footprints of many viruses that have become either silent or fairly dormant parts of our genetic makeup. And so let's leave it at that. So in between those two extremes, I can talk a little bit about why, with my own background with gain of function research, if I had known what I'm going to tell you about in a minute, I would have designed a set of experiments that are very similar to what has been done in China at the Wuhan Institute of Virology. So with that background, I'll go through a few slides and entertain your questions. So I'm going to do a share screen and go to the next slide. Whoops, there we go. All right. So that was the YouTube. All right. Before I do that, I apologize. I had forgotten. I have to minimize the this. Okay. All right, Joe or Hillary, can you tell me how to get rid of all the little gallery on the side of my share screen? Okay. Anyway, before I go on, I'm just going to point out again, there is this presentation tomorrow on the Delta variant. Sean Crody is the guy at UCSD, and that recording will be available to the public at 6 p.m. if you can't get on at noon. The other thing I want to point out is that we have a local character, Dr. Joe DeRisi, who's a professor at UCSF. And if you look up his YouTubes, you'll get an idea of the kind of research that goes into trying to track viruses that are of human concern. And he's very entertaining and a very good speaker, so you might have fun with that. All right, so.
Are you okay with the not having the gallery view? Did you find that?
I'd love to get rid of it.
Well, you should have a view button.
There we go. I just got it. Yeah,
Oh.
it was hiding behind my share screen.
Oh,
All right.
okay, good.
So here's the question. Where did SARS-CoV-2 originate? There are theories that there was an accidental release from a lab. And then the other theory, the competing theory, is what's called the zoonotic spillover. And they both, I have to point out, are viable theories. They're both in play. I don't know that we'll ever get to the point where we can say conclusively what happened. But it is of interest, obviously, some would say very important, to know where the virus came from. How did it emerge or originate? Because it affects how we work together across the globe to mitigate the risk of future outbreaks. You may or may not have watched, for instance, the Netflix series called Pandemic, which is very good. It came out in 2019 before this pandemic. And you in that series of presentations, you may learn quite a bit. I certainly didn't know about all of the efforts around the world to track where the next viruses, next epidemics and pandemics will come from. So that's another one. All right, so there are a lot of research publications, meta-analyses, which means that people are taking other people's data and putting it all together and trying to figure out on a bigger scale what's happening. There have been a lot of letters from groups of scientists and published in eminent journals, some of them arguing for the lab leak theories, other of them arguing for a zoonotic or natural occurrence of this virus. And they are excellent analyses, but nobody has complete data. This is all based on incomplete data. And then on top of that, of course, as many of you will be acutely aware, politics has muddied the waters of this question. There's been gain possible for certain political characters to point the finger and say they know what happened. I would say very seriously that nobody knows what really happened. And then this last point, if you're interested, there's an excellent Newsweek article from June of this year that was really fun to read about amateur data sleuths who have done some incredible work digging up what they call smoking guns from the Chinese data banks. And you can tell that the Chinese are very, very obviously nervous about that. As soon as the data sleuths post their findings, the data banks go dark. So it's China is acting in a way that is probably endangering its own credibility, unfortunately, in my opinion. So I've just listed here a few of the ones that are more newsworthy, not necessarily accurate, but this is an article, Newsweek, June 2nd, 2021. And the group of amateur sleuths is called Drastic, and they'll explain why they call themselves that acronym. But they have been joined by some very credible scientists and medical doctors and data analysts who have worked with them to clean up and make their reports even more legitimate. Very interesting. The next one I've listed here, the Bulletin of Atomic Scientists published an article by Nicholas Wade, I think about 2 weeks before this one, so late May. Nicholas Wade is a well-known science reporter, and I objected to his article because as soon as I started reading it, his particular bias that he believes that the virus was leaked from Wuhan was too obvious to me, and I had trouble with it, and it took me a couple of weeks to calm down and go back and reread it in detail and you know, get his the stuff that he was arguing about. So there's both these, the Newsweek and the Bulletin of Atomic Scientists, have some incriminating, potentially incriminating, I shouldn't say incriminating, but potentially incriminating information in them. So then there had been early in the pandemic, in I think the journal Lancet, a medical journal, there had been a letter from MDs and scientists saying that there is no way that this could be a lab leak, that it was definitely from a natural source and coming through various intermediate animals yet to be identified. That was mirrored in May 2021 this year, 20 scientists that were saying, I don't think so. I think we do need a good investigation. There's enough that's a little bit screwy about this virus that we really need to know more in order to know whether it was a leak from the lab, which would affect, obviously, our preparation for the next pandemic. I'd also like to point out there are some people in my research that I came across, for instance, this young woman who I believe is Australian. I saw the tag on her video and went to watch it, believing that it was a news story. But it's actually, it turned out once I listened to it and got more and more uncomfortable with it, it's really a sale for her book. And she is quite, as far as I'm concerned, very biased in her outlook. My science training says when you smell bias, you smell a rat. So I'm not very interested in her stuff. So be careful if you decide that this is of interest to you to do some sleuthing, be careful. There's a lot in it. I actually called a friend of mine who writes for Science Magazine, a science journalist herself, when I first got interested in that. And she was the one that said, you know, yeah, there's a lot in it for people who are science journalists right now to make big splashes. And so people are making claims that they really don't have sufficient basis for. All right, so let's go back to this gain of function idea. Okay, so I opened the talk by saying gain of function is what holds all of what we're talking about today together, that the generation of variants is a process which is gain of function as far as we humans are concerned, because the virus is gaining more ability to attack us, cause more concern for us. Obviously, there are changes that make it less so, but those are not as worrisome. So what I wanted to point out is that there are a few pieces that are disturbing when you take them out of context, but just to point out that this kind of research goes on all the time. It's what's being done by our government agencies, international agencies, in order to try and track where the threat for the next pandemic is. And among all of those data that people are collecting all the time, trying to track where's the next avian flu, where's the next swine flu, this one stuck out. And it is the report that, and it was published by the Chinese and picked up by an English newspaper as a page 22 article, and nobody paid attention because who cares? But there was an incident in Yunnan in the south of China where 6 men who had been shoveling bat guano in a cave, probably to send to the United States as an ingredient in garden fertilizer, 6 men became ill, quite ill, and 3 of them died. And those 6 people were treated in Chinese hospitals. Samples were taken. A lot of work was done to try and figure out what was this disease. It's an unknown disease. What is the infectious agent that was responsible for their illness? The fact that it was a 50% mortality rate, extremely concerning. So the incidence is called, and I've lost my cursor, I don't know if you guys can see it, but let me go backwards here, was in what's called the Mojian, I'm sure I'm making a mess of that pronunciation, but the Mojian cave. And when I heard about it, I thought, okay, what would I do if I knew that 6 people had been infected and 3 died? So obviously you would start by studying those 6 patients, hoping to identify the fatal agents. That's what was done extensively. Part of what was found by those amateur sleuths were master's degree and PhD theses written about those 6 cases of disease in China. And they discuss an awful lot of what was found out about those people, but in no instance was a fatal agent identified. So what do? So you go back to the cave and you continue collecting samples. You've got to find what is this lethal agent that is so infectious and lethal. And so how do you do that research? Well, the way you do that research, you're asking the question, I've got this little sample of dirt or bat guano or whatever it is. I'm taking it down into different pieces, and I have to put it into human cell culture. I have to put those samples onto human cells in culture, and then eventually into mice that have been engineered in order that the mice carry the ACE2 receptor, because I'm asking the question, where is the element that can kill the human cells in culture and that can also attack and cause disease in the animals that are carrying this ACE2 receptor? In that process, you are asking, do I have an agent from the cave that already has the ability to cause disease and death in humans? However, you also have to admit that by doing that, you are asking for gain of function from the sample that you've put into culture. You also want to see, can these samples, if you passage them, that is, you put them onto human cells, they may not kill the first set, but you take the viral progeny from that and you put it onto the next one. Basically, what you're doing is you're giving your human cell culture an infection, and the variants that are generated as those viruses replicate can gain the function to kill the human cells. And so you're asking, does it take 20 passages on human cells for them to become lethal? Does it take 2 passages on human cells for them to become lethal? Does it take 2,000 passages on human cells before those viruses become lethal? So you can see you are actually in the process inherently. There's no avoiding it. If you want to know the answer, do these samples have the potential to cause a pandemic? You are going to be generating samples that could become the next pandemic. I hope that makes sense to you. So I'm not sure that I need to go through all of this again. There's a little repeat at the top. The Mojang cave, 6 cases, 3 men die. The way in which they were worked with was quite legitimate. The gain of function, as I said, will happen. The gain of function also is for the bat coronaviruses, was developed in the University of North Carolina by Richard Barrick, a United States researcher, and he trained the Wuhan Institute of Virology to continue that work. But they are using the technology that was developed in the United States in North Carolina to do this work with, for instance, the samples. So the questions that I have, for instance, here's a fact. A fact is that in 2014, the United States funded the research at the Wuhan Institute of Virology for 5 years. So 2014 through 2019, they had, I can't remember the size of the grant, maybe $6 million that was funded via an alliance whose home is in the United States. Peter Daszak is the virologist who's the head of the Eco Alliance. So they have, and I looked up that grant and read the grant, and sure enough, that's exactly what they're doing. They're looking for the smoking gun from the Mojang cave. So 2014, they get funded. Also 2014, China makes a contract with France to build the safety laboratory of level 4, very high level of safety in China. And I would think that both France and the United States, at the bare minimum, knew what was going on in China in 2012 that motivated them to go ahead and do this funding. So here's France is helping make that facility. Anyway, so I mentioned that there's lots of publications that came out of the Wuhan Institute of Virology, as well as less polished ones, which were master's theses, PhD theses. And as I said, the virus was not readily identifiable, but in the process, at least 8 viruses were uncovered in those studies. They're very similar to SARS-CoV-2, but they are not SARS-CoV-2. So this, I don't expect anybody to read. Hopefully it'll be in the recording, but it is part of the information that was uncovered by this team of amateur sleuths. So I'm going to zip over this. This is also too much detail for here, and just go to the end. You'll have this in your recording. There are quite a few people that if you get interested, you can track down. Any one of these guys is very interesting to look into their research, their writings, what they have to say about the whole thing. This is very interesting to me. This is an Australian woman who worked in the Wuhan Institute of Virology. She is a bat coronavirus trained Australian scientist, and she was there for several years. And her testimony is that the accusations that people are making that say this was a lab leak or that it was an escape from the lab, she says this is really, she can't say 100% it didn't happen, but she said it's very much, very, very unlikely. Her own experience in that laboratory, working there for quite a long time, before she left in 2019. This is the latest, excuse me, that I could find article on the whole issue of where did it come from. So, there's a link in here to a review. I think I have the review itself on the next slide. But as I said, this is an ongoing discussion, let's put it kindly, a discussion about where it came from, whether it was a zoonotic origin or a laboratory incident. And this is just a review of what's in that article. The conclusion is that the genetic analyses of SARS-CoV-2, where people have been claiming there's a mark of it having been gene edited or lab engineered, they are refuting that. They also point to the fact that there are there are a number of studies, very reputable studies of blood banks where the samples were collected long before the pandemic. And those blood banks, many people believe, contain antibodies against the SARS-CoV-2 virus, showing that it was circulating in Europe, possibly before the summer of 2019. Another thing is that even though there is no intermediate animal for this virus to get to humans, there are records of civets and raccoon dogs, which are possible intermediaries, that they were sold in Wuhan wet markets mid-2019 and could explain the evolution of the virus from no, it can't infect humans to yes, it can. Third or fourth point is that they had a stronger than usual flu season in Wuhan area that could have masked the appearance of SARS-CoV-2. And the last one is that when viruses do emerge zoonotically from the animal world into our world, they are not lethal to the level that they get to once they've kind of acquired the ability to grow in humans, that they've made enough of a foothold that they can infect us, replicate, and gain variants that are gain of function variants. And then they cause a pandemic. So I hope that's the end. And I'm going to do my stop share and see what we've got. So that was a whirlwind tour about a very complex, very complicated, fascinating discussion among virologists, epidemiologists, medical doctors. All kinds of people are weighing in. And unfortunately, as I said, politics is coming in and making it difficult to even make a scientific judgment about it.
We do have a question from Roger. Roger, do you want to ask your own question?
Okay, thank you very much for your presentation. And it's been about the vaccine development, but it raises the question to me about therapies. And is there anything in this research which contributes to therapies for the infection?
Absolutely. So, as you know, there are a lot of efforts for antivirals. That's one aspect of therapies, antivirals to prevent the interaction of the virus with the cell surface. So there's a lot of attention to various aspects of the virus surface to basically block its ability to infect. That's one end. And then, of course, the ACE2 receptor is an angiotensin 2 pathway element. And so breathing, regulation of hypertension, all of these things are affected, which is part of why we get the horrible symptoms that we do when we're infected, because it's an interruption of those regulatory pathways. So people are working hard at trying to not only balance those effects when they see a patient in the clinic, but also use that information to understand how to interrupt the cycle, lessen the amount of damage to somebody who's infected, and or prevent the damage at all. So a very interesting one, if you get really interested, is that there is a I call it a goop. When I was doing vaccine development for agricultural use, there's a goop called ivermectin, and ivermectin is used extensively in some especially tropical third world countries to prevent things like river blindness and liver flukes and things like that. And so there have been some studies, some epidemiological studies that show that those populations that use ivermectin as a kind of a standard, you just take it with your cereal in the morning because you know you're exposed, that is an explanation why Africa has not been hammered, for instance, because the countries that use ivermectin regularly, ivermectin, some people have shown, will interfere with spike ability to interact with the cell surface. And so it has an antiviral effect. So there are these amazing out of the blue things that are coming up, but you can bet your bottom dollar that every big company like Merck and Sanofi and Genentech are working very hard to try and make a good buck off of this pandemic by developing a good antiviral. Yeah.
So if, for example, if somebody came to the hospital with a case of COVID, would it help them to give them an injection of the vaccine?
So I think that would be certainly part of it, but I'm backing off for a couple of reasons. First of all, if you're coming into the hospital, you're not in very good shape. And so your immune system is already in haywire. You have to be really, I'm not an MD person. I listen to these presentations about how much struggle, especially, for instance, we have some amazing people in the Bay Area. For instance, regularly on the UCSF Department of Medicine grand rounds, there are two doctors from what used to be called San Francisco General, the now called Chan Zuckerberg Hospital, who are HIV specialists. So they've been working with immune compromised people for decades. And how do you protect people who are immune compromised? And there's many people, especially elderly people, who are immune compromised. How do you work with them? You can give them a vaccine, but they won't respond to it in a way that you or I would. So I'm not going to even try to answer a question about would you give somebody a vaccine when they showed up? I would think it would be a pause button because you already have a pretty stressed situation on your hands. You're not necessarily wanting to redirect the immune system in that moment.
Okay.
I don't know. Anyway, there are some, all of these UCSF broadcasts are available to you. You can go on, there's at least 2 sessions on immunocompromised people. The early ones that you'll see on that website that Hillary will send you the link for, a lot of them are about evaluating remdesivir, about evaluating the various different candidates for therapeutics, but the medical people were really hamstrung at the beginning because there was nothing that they could find that was effective.
All right, do we have any other questions? If not, I would,
Excuse me.
yes.
I wanted to first thank you so much, Irene. I was pleasantly surprised to understand your talk. So that was really great. But you also hinted at something, and I don't think I'm the only one who would be interested in that's learning more about the work being done in cancer vaccines. It affects many of us here today.
Yep. And actually that's closer to I'm a cancer cell biologist. So one of the things that I feel really quite silver lining-ish about is that to move these cancer vaccines, so for instance, BioNTech company started by a Turkish couple in Germany, that was their goal in life, was cancer vaccines. Then the pandemic comes along and they realized that they could be very helpful by pivoting and taking their experience. They went to Kaiser, and Kaiser had the vaccination development and ramp up knowledge to make a good partnership with BioNTech. So it looks as though the fact that the Moderna and Pfizer worked so well, it catapulted that technology forward.
Oh, great.
So that's a separate presentation that I've done for other groups. It is exciting. There had been clinical trials started. Hopefully they're getting a push from what we're learning about how these agents work in the body.
And is that particular cancers or just more of a general?
So in general, the cancers that are first up when it comes to clinical trials are generally the ones that are most quote unquote popular, the most likely. Colon, lung, melanoma is a big one because it's susceptible to antibody therapies, et cetera, et cetera. And yeah, leukemias, lymphomas, the things like ovarian cancer, unfortunately, not enough incidence to really get a lot of traction. Anyway,
Mhm.
it's a whole field. Yeah. So,
Thank you.
yeah.
So with that, Irene, you've been very generous with your time today, and I want to thank you for that. It's also interesting to see a presentation which is so engaging and yet is still so complex, and you have a real gift for explaining it very clearly.
Very kind of you. Thank you.
And so I want to thank you and everyone for joining us today, and bravo to you.
Thank you.
And we will once again convene next month for further presentations. And Irene and all of the people who are guests today, feel free to join us again. If you want to be put on our permanent mailing list and you are not an Ashby Village member and already signed up for this group, send me an email and we'll put you on, and so you'll automatically be notified of any future and all future meetings on the second Wednesdays of every month.
Thank you, Joe and Hillary, for hosting me. I really appreciate it. And thank you, everybody, for coming.
Thank you.
Thank you.